have Dr. Lynn Barber. Lynn is a tenured professor in endocrinology, metabolism and diabetes. She's very accomplished and known worldwide actually for her work in fetal impairment, lifespan, growth, obesity, diabetes, predisposition in utero. She's been NIH funded for over 20 years. She was awarded the ADA 2018 Norbert Frankel Award for her research, teaching and clinical contributions to diabetes and obesity in pregnancy. She has mentored over 25 MDs and PhDs really well because they've all gone on to be incredibly successful. She was on the editorial board for Diabetes Care and is a past chair for the ADA Scientific Planning Committee for Pregnancy. She served on several NIH panels, and she is currently on a joint Endocrine Society, European Endocrine Society Guideline Committee for Diabetes and Pregnancy. So with that, I will pass the floor over to Dr. Barber.
Thank you for that very kind introduction. I want to call out two people before I start. My co-principal investigator for the last 4 years who hasn't ditched me, Terry Hernandez, Dr. Terry Hernandez, and Janine, you'll see at the last slide, has really made so many of these studies possible with both her expertise in the bionutrition, CCTSI, and just for the tremendous scientist she is. So anyway, thanks ahead of time for that. So we're going to pivot quite a bit from animals and infection to mothers, children, and the next generation. If I can make this go forward. I have no disclosures. And this audience is probably quite aware that unfortunately 2030 is coming soon in 7 years. It's pretty sobering to think that 1 in 2 adults will be obese as a projection, 50% of our population. Class 3 will actually be the most common BMI class in women, and 1 in 4 of our children, 5 to 9 years old, to be obese. So there's a lot of interest in what we call the DOHAD hypothesis, developmental origins of health and disease, trying to understand what are some of the earliest features, what are some of the earliest insults that may predispose to this, because we're obviously seeing some of these incredibly early in life, as well as type 2 diabetes as early as 3 years of age, which I'll show you. So our group for the last 20 years really has been very interested in trying to understand the metabolic factors in the intrauterine environment that can have these profound effects on development and then enhance susceptibility to later metabolic disease, what we call DOHAD, which is truly no longer a hypothesis. Started out with animals, but after those rats bit me too many times, we've been switched over to humans for the last 20 years. And that's what I'm going to be showing you a little bit of some of this research that we've been doing. You know, we know that with diabetes, and with gestational diabetes, that results in very, very high fetal insulin levels when that glucose crosses the placenta. And very high leptin levels actually changes the fat cell development number, as well as the size of the cells. It causes enlargement of the fetal pancreas, the heart, changes the nephron number in the kidney, and also causes increased liver fat. We also know that high fetal insulin leptin levels actually affect appetite regulation in the hypothalamus. And one of my very important co-investigators over the last co-PIs over the last 20 years, Jeff Friedman, has shown with the Oregon Animal Primate Project, if you feed a mother monkey a high-fat diet, you will actually create non-alcoholic fatty liver disease in the baby monkey. Mitochondrial dysfunction, appetite changes, and behavioral changes are really actually pretty spooky. We know that energy expenditure and mitochondrial oxidative capacity are also affected in humans, and this can increase the risk of glucose intolerance in childhood. And remember, we inherit our mitochondria from our moms. My daughter reminds me that all the time. We also know, the focus has really been on diabetes, gestational diabetes, but in fact, maternal obesity is a stronger risk factor for childhood obesity than even gestational diabetes. We know that from Pat Catalano's data, that it wasn't GDM that predicted childhood obesity, it's actually maternal BMI. We also know that birth weight is a really terrible surrogate for fat mass or a predilection towards childhood obesity later in life. That in fact, it's percent fat mass at birth that correlates with the development of childhood obesity by a DEXA at 9 years of age. So we've been really focused on only diabetes, and in fact, obesity is what's staring us in the face now, especially if it's going to affect 50% of the population. And Kristen Boyle's done really elegant studies showing that if you take the mesenchymal stem cells from the cord of offspring that are born to women with obesity, that they actually have much greater adaptogenic potential. They refer to differentiate into adipocytes and skeletal muscle cells, compared to those mesenchymal stem cells in the cord from offspring of normal weight women. So our group for the last 20 years with Terry as co-PI for almost all of this have really been trying to better understand this whole process in utero, exposure, and then later programming. We call our group, there always has to be an acronym, Infant Gold Investigations and Gestational Origins of Lifetime Development. And we've been fortunate to be funded for the last 20 years. I'm going to show you a little bit of some of this data. Dr. Hernandez will soon, we just sent a paper on a randomized controlled trial in gestational diabetes that will be coming out. I'll show you a little bit of some of the data that's informed our current study that is ongoing right now. So the first study that really helped us really kind of rethink about this whole process, what we call the fetal programming study, that we started back quite a long time ago. What we really wanted to do was to try and tease out the independent effects of maternal obesity, maternal body composition, carbohydrate and lipid metabolism on fetal fat accretion and neonate adiposity. And once again, we use DEXA because, I'll just point because I'm not good enough with the computer. This baby doesn't weigh as much as this baby, but you can see that this baby has almost twice as much fat. So you really want to look at body composition, not birth weight, or what we call PPOD, air displacement plethysmography, to really look at that body composition. One of our first studies, Janine will remember, is we wanted to really focus on obesity. So we looked at, what are the difference between normal weight women and obese women? Why are obese women having these babies that are with higher fat? And so we gave normal weight women and obese women eukcaloric diets. And then after that liquid diet, we studied them over 4 hours, 10 times, measured everything you can imagine metabolically. And what was interesting is we found out that our women with obesity, even early in pregnancy, we did this at 16 weeks and at 28 weeks, already had much higher insulin levels than women who were normal weight. They didn't have GDM. They were incredibly insulin resistant already. And their glucose levels were high. These crazy high insulin levels that they had actually suppressed free fatty acids. And so there wasn't too much of a difference. But what was really interesting to us was this triglyceride story. So obese women, triglycerides also go up in pregnancy, probably partly due to estrogen. But obese women were already starting out with 30 to 40% higher triglycerides than our normal white women, which continued all throughout pregnancy. And we could actually capture this whole 4-hour area under the curve with these 10 time points by a 1 or 2 hour postprandial triglyceride. And when we actually looked what predicted newborn adiposity, it was the triglycerides, not the glucose. And in fact, glucose added absolutely nothing to our regression model. And we found out that the best predictor for percent fat by DEXA was a 2 hour or 1 hour, 1 or 2 hour postprandial triglyceride early in pregnancy, not later in pregnancy. Glucose was not an independent predictor. And so Terry and I were looking at the data, trying to see, wow, so can we predict what babies are going to end up with excess fat? We noticed that there was this display in triglycerides in pregnancy in terms of these women starting out. Not all these women with obesity had high triglycerides. About half of them started out with triglycerides fasting less than 120, and the other half greater than 120. And interestingly, what we found out is this 2 hour postprandial triglyceride prediction was not very strong, with a correlation of only 0.39 in the women who started out with lower triglycerides. But in the women who started out with fasting triglycerides greater than 120 at 16 weeks, that R was 0.95. 0.94 in terms of the 2-hour postprandial triglyceride early predicting adiposity. We also found this interaction between triglyceride and glucose later in pregnancy, but not early. And remember, the fetus can respond to this high glucose exposure at about 14 to 18 weeks. That's when it really starts ramping up its insulin. So we hypothesized that maybe high glucose caused fetal hyperinsulinemia later in pregnancy, and you combine fetal hyperinsulinemia, which is a growth factor, with all this triglyceride availability, and maybe that's what was really setting the stage for increased fat. And again, Terry and I have been really, over the last 20 years, been looking at normal weight women with obesity, women with gestational diabetes, especially those with gestational diabetes, GDM, who we can control with just a diet, who are not on insulin, not on any treatments. In the OB world, really, the only population that we really focus on to try and prevent fetal overgrowth is in women with gestational diabetes or pre-existing diabetes. We kind of close our eyes to obese women. And we pretty much just look at the glucose. We're not looking at any other nutrients. And what Terry, when she started analyzing our 24-hour continuous glucose monitors, so we put 24-hour continuous glucose monitors on normal weight women, obese women, and women with GDM, the GDM women and the obese women in the green and the red. And we control for their diets, as Janine has helped us do. And you can see that the obese and the GDM women look pretty similar. Not only do they look similar in their glucose, but they also look on the right panel very similar in their triglycerides, much higher than normal weight women. But we don't target GDM. We don't target women with obesity. We only target women with gestational diabetes. And we're not only interested in fat deposition in subcutaneous tissue in these babies, but what about liver fat? We know that non-alcoholic fatty liver disease occurs in 40% of children with obesity, and it progresses much more rapidly to NASH. And so David Brumbaugh, who's a pediatric gastroenterologist, said, really, let's take a look whether any of these babies are born with more fat in their livers. So designed a way to put a two-week-old in an MRI and magnet. And we decided we would look at the extremes. We'd look at offspring born to women with both obesity and GDM compared to normal weight and see whether their 2-weekers had any evidence of fat in their liver. And in fact, these babies born to the mothers with obesity and gestational diabetes had 68% more fat in their liver, pretty much at birth, 2 weeks of life. And it really looked like it was driven by the obesity. It was really mainly the women with obesity that showed this. And then we've been doing a randomized control trial in gestational diabetes, challenging the conventional low carb, higher fat diet with this choice diet with higher complex carbs, low fat. And in a smaller population of these women, we also had the babies go into a magnet at about 2 weeks of life. And we also found this relationship between mothers' maternal triglycerides and fat in these babies' livers in a small sample size. And what was interesting again is we saw this relationship earlier in pregnancy, soon after the diagnosis of GDM, but not later. Well, it turns out that fetuses really don't make much in terms of subcutaneous fat stores until 28 weeks. So these babies are exposed to a high lipid load before they can deposit that fat in their subcutaneous tissue. Our hypothesis is it may go to the liver to be stored. And you think, well, how do those triglycerides ever get to this baby? It turns out that the placenta have lipases just like our fat does. And Margaret Heerwagen, who is a postdoc who was working on her MD, worked with us, and we thought maybe there's something about the activity of the lipases that end up hydrolyzing these triglycerides to free fatty acids, because that's what lipases do. And maybe then there's more availability of free fatty acids for fetal fat accretion. And she actually showed that the activity of lipoprotein lipase, so you have to use a fresh placenta to get the activity, of course, actually correlated very strongly with adiposity by skin folds at birth. So this is probably some of the way those triglycerides then can be hydrolyzed to free fatty acids and become available to the baby for deposition in the liver or in fat stores. So our current study we really want to tease out this relationship between fasting and postprandial triglycerides in women with obesity, in terms of how this may cause increased deposition in subcutaneous fat in the babies as well as liver fat. Problem is, up until recently, how do you check fasting into our postprandial triglycerides? In gestational diabetes, we sent all our mothers to glucometers, and they check their fasting in 1 or 2 hour postprandial blood glucoses. And it turns out, fortunately, there's an FDA-approved triglyceride point of care meter that we have validated in pregnancy, and we're going to be using this to check triglycerides almost like you would your glucose with a glucometer. And so our specific aims is we wanted to test the hypothesis that women who are overweight and obese, that maternal triglycerides will predict newborn percent fat in both boys and girls. We've ramped up the numbers that we should have the power to look at boys and girls because boy placenta and girl placentas are very different in the risk of boys going on to develop this metabolic syndrome more than girls. And it's going to be independent of maternal glucose, mother's insulin resistance, or BMI, or gestational weight gain. But we think that glucose may moderate that fact later on when the baby really starts responding with that hyperinsulinemia from the elevated glucose. And in addition, that ends up causing storage of this fat. And then our second aim is just to look at what's going on in the liver. And we postulate it's going to be early exposure of these triglycerides that really correlates with liver fat before the babies develop their subcutaneous tissue that maybe ends up being stored in the liver. And then our exploratory aim, which is a group of incredible co-investigators, placental experts, lipidomics experts, and the mesenchymal stem cells that I showed you with Kristen Boyle, will evaluate the effect of maternal triglycerides on placental pathways associated with lipid transport, metabolism, on maternal and cord blood lipidomics, and then on newborn umbilical mesenchymal stem cell adipogenic potential. And the question is, great, what can you do about it, right? You really can't give, statins to pregnant women. What you can do is you can give high doses of omega-3 fatty acids. And omega-3 fatty acids, as I'll show you in the adult literature, does decrease triglycerides. So we are hypothesizing in a pilot study where we're doing only with those women who have triglycerides greater than 120, we're randomizing them to very high doses of omega-3 fatty acids, 4 grams a day, which is what's used in the adult population to lower triglycerides, versus a placebo, which is soybean oil, which I'll show you. And we postulate that we can normalize those high triglycerides, so they're more like the normal weight women, and then possibly decrease the risk of excess fetal fat deposition. And then the exploratory aim is we're going to look at the placentas and do all the same things. And again, the rationale behind this 4 grams of omega-3 fatty acids comes from the adult literature, in which Labaza a number of different omega-3s with both EPA, high doses of EPA and DHA will lower triglycerides by anywhere from 30 to 40%. And then we're hoping that we can use that to see whether we can normalize the triglycerides. The formulation we're using is a formulation called Nordic Naturals. It has a very bioavailable DHA, EPA content. Every capsule is about 1 gram, which is a lot. These women are taking 2 capsules twice a day. Terry and I both taking it. It doesn't taste like fish oil. And it's actually pretty easy to take. And then our placebo, the company made an exact placebo, identical placebo of soybean oil. And it kind of tastes a little lemony because both of them have it. And it really isn't too bad, I don't think, for pregnant women. And fortunately, you're gonna think, well, these women are gonna take that, you're not gonna see anything, which could be true, but we do have a way to quantitate the compliance of whether they're taking their omega-3 fatty acids by working with Bill Harris, who's a lipidologist, who developed this omega-3 quant index. It's like an A1C for fat. So 1 drop of blood, you can see all 24 fatty acid species in a red blood cell membrane. So you could tell what someone's been taking over the last 3 months in terms of the omega-3s being incorporated into the red cell membrane. And so this is the schematic for our studies. We just started them. We lost a year with COVID. So we're just starting our second year. So in both studies, we're taking pregnant women, recruiting them in about 12 to 16 weeks. The bassinet trial is the one that I told you with the point of care meters where we're not treating them. The TOTS trial is the one where we're randomizing them to the omega-3 fatty acids versus placebo. And we're doing all these baseline labs. We're giving them a triglyceride meter. We're also giving them a glucometer, replacing a continuous glucose monitor because it's so important to figure out, any contribution from glucose. And they're checking their sugars and their triglycerides 4 times a day, just like you would ask a woman with diabetes to check, fasting to our postprandial. They're doing that for 4 days, and they're doing it 4 different times during pregnancy. So we should have, for the first time, 64 fasting and postprandial triglycerides during the course of pregnancy, along with glucose. And no one has actually done this. The triglyceride data out there is just, on a couple of fasting samples usually. And so we're doing that at all these different time points during pregnancy and at delivery. Our wonderful lipidomics experts, Brian Bergman, we're looking at maternal blood lipidomics. We're going to actually take it 2 hours before the baby's delivered and then look at the cord blood and see kind of what's getting across, what's not. We'll actually be able to do that omega-3 fatty acid index not only on mom, but on the cord blood to see what the baby's getting. We'll be looking at the placenta. We'll be doing body composition by PPOD at birth. And then we're bringing the kiddos back at 17 days to look again at body composition by the PPOD as well as NMR for their liver fat. And we're very fortunate to have met just a wonderful MRI physicist on campus here who's helped develop this M-Dixon quantitative method where we can actually quantitate the liver fat of these babies in about 15 minutes. And the baby on the lower panel actually has more liver fat by lighter blue than the baby on the upper panel, about twice as much liver fat. So again, we're using PPOD and this way to measure fat by MRI for our outcomes. And I was just going to finish up and show you a little bit of just the first 18 mothers who are starting to go through this. Actually, our recruitment has actually been surprisingly quite good with our amazing research team. In terms of display of their triglycerides, so this is called the bassinet study. And this is a diagram of about 200 fasting triglycerides by these point of care, both at 16 weeks and then all through pregnancy to 35 weeks. Once again, we see this big display in fasting triglycerides in these women who are obese. So they look like they have the same phenotype, but by their triglycerides, they have very different phenotypes. Some of those triglycerides, are as low as 65 fasting. Some are as high as 210. And at 35 weeks, they range from 175 to 325. This is about 600 postprandial triglycerides taken at breakfast, lunch, and dinner. Once again, you see this big display. So we really want to see, it's not all obese women that have babies that are big, and maybe we need to be focusing on the ones with higher, we'll see. And so in conclusion, what we're really trying to do to challenge the paradigm of only focusing on glucose in diabetes and gestational diabetes, and really shift the focus to women with obesity and lipids, which is going to be 50% of the population in a very soon time. I think we have good preliminary data showing that fasting and postprandial triglycerides in obese moms are about 30 to 40% higher than normal weight moms, and they may contribute to excess subcutaneous fat and liver fat. And by our point of care triglyceride meter, we should be able to really establish that relationship. We think it's going to be the women who start out with higher triglycerides that will have the strongest relationship with adiposity. We want to find out if that relationship is continuous versus there's maybe a threshold effect, and that this threshold may target women. We think that the higher glucose levels later in pregnancy will result in that fetal hyperinsulinemia. So then you have a perfect storm of high fetal insulin levels with all this triglyceride delivery for fat deposition. We think that for the liver, we're going to see the relationship between triglycerides and liver fat deposition earlier in pregnancy before the baby has developed its subcutaneous stores, so that exposure of those triglycerides ends up going to the liver. And then lastly, in women with obesity and high fasting triglycerides earlier in pregnancy, they actually targeted for this 4 grams of omega-3 fatty acids to normalize the triglycerides prevent excess fetal growth for our TOTS trial. And so this is just a paradigm. Dr. Terry is hugely helpful in all of this that we developed for a talk I gave at the American Diabetes Association. It's more complicated than just glucose in women with diabetes. We think that mothers come into pregnancy with their genetics or epigenetics, many of them obese with pre-existing insulin resistance. We've shown that. Decreased beta cell reserve and metabolic syndrome, often they don't eat very well. We've also shown that human placental growth hormone in animal models is probably the most important hormone to cause insulin resistance. So then they hit more insulin resistance at pregnancy, they may gain too much weight. We've shown that in their muscle, they're insulin resistant, so there's more glucose availability to the placenta. We've shown in their adipose tissue that they're more insulin resistant, increased free fatty acid delivery. And they have more chylomicron TGs from their gut, and their liver generates more VLDL triglyceride. And then the placenta always is the deciding factor of what gets across and what doesn't. So behold to the placenta, who has more hormones than any other organ in the human body. So as an endocrinologist, I bow to the placenta. It's very difficult to study, so we have wonderful investigators who are studying the placentas with us, Thomas Jansen, Teresa Powell, Kartik Shankar. And we know that it has its own placental hormones, it has growth factors. The sex of the placenta can determine actually nutrient sensing. Its metabolism and its transport obviously are going to result in what ends up crossing that this fetus sees. And then the fetus is then exposed to increased glucose, which causes fetal hyperinsulinemia, increased triglycerides. It can be deposited in the liver, it can be deposited in the subcutaneous tissue. But then the newborn's genetics and its beta cell response are going to have a lot to do with what happens in terms of its appetite regulation, its behavior, mitochondrial function, stem cell differentiation, energy balance, and I haven't even talked about the microbiome, which is incredibly important. Jeff Friedman is really taking a lot of that part of the study and evaluating. It's very important. And then postnatally, we can give another hit to this baby. We know that babies develop their fat cell number pretty much for the first 6 months of life, and they actually double or triple their fat cells. And so if they're overfed, if they have rapid weight gain, that's another insult in terms of it, and changes in their microbiome. And this could lead to childhood obesity, non-alcoholic fatty liver disease, metabolic disease. If it's a girl baby, she can become a mother, and the whole cycle continues. So I want to thank. None of this would be possible with our extraordinary team science. These are just the people that have been involved in this study presently. I mean, I would have just been the whole time throwing out names, especially the Dr. Hernandez who's sitting there, Jeff Friedman, who's actually the director now. He left us and went to Oklahoma, but we're actually collaborating on this top study, doing a lot with the microbiome. A whole bunch of just brilliant investigators that we're so fortunate to work with. Our amazing research team that you see in yellow, the adult and peds CCTSI, perinatal research team led by Jocelyn Vipers, our lipidomics score with Brian Bergman, our labor and delivery nurses who put up with us with these women coming in the middle of the night and us getting their placentas and their cord blood and everything else. Janine, for which none of this would be possible. And Ron Sokol on the CCTSI, our funding, and of course our mothers and our babies. And thank you.
It's really great. It's fascinating work. Thank you. I'm very interested in one of the most important determinants of child onset wheezing is maternal obesity and asthma. I'm a pulmonologist.
Yeah.
But I study obesity and asthma, so some of the things that relate to increased maternal leptin and insulin cause airway parenchymal uncoupling, and these kids are more bronchial hyperresponsiveness. So it'd be nice to look at kids' lungs as one of the end organs that seem to be impacted by these metabolic derangements.
Yeah, let me know.
Because if you can do it with, using impulse oscillometry, you can do it, without any significant effort.
Yeah, that'd be very interesting. Please reach out.
We'll do.
Yeah, I think it's very interesting. I have another question for you.
Yeah.
My understanding, and one of the reasons I run the high-risk OB clinics, and I always tell our obstetricians to quit giving the folate after the neural tube is developed, which is done by about 8 weeks. Because of this relationship with possibly folate and asthma reactivity and atopy being a methyl donor, is there still any,
No, I think it's sort of changed completely over time. And it should, because that increased methylation can lead to increase for allergy and atopy. I don't know if that's a mechanism related, it's more related to allergies than I think
an allergy,
asthma,
right.
but it's certainly one of the factors we looked at.
Yeah, great. All right, well, great. Yeah, you know how to get a hold of me by email.
Yeah, well, I will send you an email.
All right, great.
Thank you so much, this is great.
So we're out of time. So if anyone else has a question, please ask Dr. Barbour directly, and thank you all for coming.